Okay, so welcome back to this next video in which we are discussing the ionotropic glutamate receptors. Okay, right, so we've finished now our discussion of the AMPA receptors. What we now want to turn our attention to is the kainate receptors. Okay, so remember initially I told you that the kainate receptors are often abbreviated to KARs for short. Okay, and this stands for Cannic acid, which we'll come to. It's an agonist, a selective agonist for the kainate receptors rather. And then R for Cannic acid receptors. Okay, right, so again, this is another family of ionotropic glutamate receptors. So their ligand is glutamate. They all have this tetrameric structure where the subunits have that characteristic wherever it's gone. They're all tetramers, not like that, like this, okay, and each one of the subunits has this characteristic structure. Okay, so we want to start off with how many different subunits do you have for kainate receptors? Okay, well you have 5 different subunits for kainate receptors. And now, in the new nomenclature, these are called gluK1, gluK2, and I'm going to write them all out because I want to tell you about their other names as well. gluK3, okay, and then it goes on, gluK4 and gluK5. So I hope you can appreciate that the new naming system is very nicely logical. Okay, so glu for glutamate, K for kainate, and then we've just got the 5 different kainate receptor subunits, okay, labelled 1 to 5. Right, so all of these subunits will have different sequences of amino acids from each other, okay, but they will all have that same overall basic structure, that same membrane-spanning topology with the amino terminal domain and the ligand binding domain and all of that, and they will all be able to tetramerize together to make an ionotropic glutamate receptor. Okay, right. So let's now discuss the old naming system for these subunits of kainate receptors. So remember, in the old naming system for the subunits of AMPA receptors, we had gone from GluR1, GluR2, GluR3, all the way up to GluR4. Basically, you continue that naming system on for the subunits of the kainate receptors, okay? So gluK1 becomes GluR5. gluK2 becomes GluR6, gluK3 becomes GluR7. Okay, so you might think this isn't so bad, but then gluK4 and gluK5 are called something completely different. They're called Cannic acid 1 and Cannic acid 2. Okay, so these names are the old names. Now, why am I telling you this if they're the old names? If you read primary research literature on glutamate receptors, people still use the old names for the subunits of ionotropic glutamate receptors. Okay, people get stuck in their ways and don't want to have to change their naming system. They don't think they ought to have to change their naming system. But the new naming system is simpler, it's better. Okay, so this is the one that I would recommend you use, but be aware of the old one, because if you're reading primary research literatures, you don't want that to be the thing that confuses you. Okay, right, okay, so let's now talk about how we can build tetrameric kainate receptors out of these 5 kainate receptor subunits, and I will reiterate what I said when I was discussing the AMPA receptors. You cannot join kainate receptor subunits to AMPA receptor subunits and make an ionotropic glutamate receptor. There is no linking ionotropic glutamate receptors between the 4 different families. Okay, so you can link kainate receptors that are in the same family to some extent, but you can't go beyond the different families. Okay, or at least nature doesn't seem to go beyond the different families. That's why we group them into these 4 families, because there's some logic behind it. Okay, right, so here is our kainate receptor, let's say. So here it is, it's this tetramer of 4 separate subunits all joined together. Right, so how can you actually build a tetramer out of these 5 different subunits? So let's begin by discussing the homotetramers which form, because those are the simplest conceptually. Okay, so remember in a homotetramer, what happens is you take 4 identical subunits and you join them all together to make a tetramer, basically. So in this slot you'll put that, well, let's take an example. Let's say we want to build a gluK1 homotetramer. What we do is we create 4 gluK1 subunits. We'd slot one in here, slot another in there, and another in there, and we then end up with a gluK1 homotetramer. Okay, so which of these Canate receptor subunits can form homotetramers in this way? Well, 3 of them are known to. Okay, so gluK1 can form a homotetramer, gluK2 can form a homotetramer, and finally gluK3 can also form a homotetramer. So all of the first 3 can form homotetramers, okay, where you have 4 identical subunits making up the overall tetramer. Okay, now let's talk about heterotetramers, which is more complicated. Now again, the heterotetramers which form within Canate receptors, thankfully, are all diheterotetramers. Okay, so that means that you only have 2 different subunits within the tetramer. You don't go wild and pick 4 different subunits and stick them together. You only pick 2 different subunits and make a certain number of each of them so that the total number that you have adds up to 4, and then stick them all together. Okay, so which diheterotetramers can you then make? Okay, so I think I might go up onto this little space here so that it's all on the same page. Right, so this is the basic rule for making a diheterotetramer. Basically, what you start off with is you need to pick one of these 3 up here. One of the gluK1, gluK2, gluK3. Okay, so if you want to build a diheterotetramer, I'm going to tell you how to do it. Firstly, you need to select one of the subunits from these 3 here that have now been highlighted in pink. So you pick one pink one. Okay, you then also need to select one from this turquoise portion here. Okay, so you'll also take you'll also pick one of these turquoise ones, and basically you can then build heterotetramers that way. Okay, again, the stoichiometry is not known, i.e. how many, well, I don't think it's known anyway, how many of each specific subunit you have isn't known, basically, and it's certainly not known how you orient them relative to one another. Okay, so the rules are this: you pick one of the pink ones and you pick one of the turquoise ones, and then you can build heterotetramers like that. So for instance, let me do an example. I want to build a Canate receptor. I'm going to pick as my purple type gluK1 and as my turquoise type gluK4. I can now build you a heterotetramer which is gluK1, gluK4, and this is going to contain both gluK1 subunits and gluK4 subunits. How many of each? I can't say. It might be 1 of these, 3 of these, 2 of these, 2 of these, 3 of these, 1 of these. How they're going to be oriented, again, I can't say. But I can build you a ionotropic Canate Glutamate Receptor out of these 2 Canate receptor subunits. Okay, similarly, I could have picked gluK1 and gluK5, or I could have picked a different one of the purple ones. I could have picked gluK2, and then I could have paired that with either gluK4 or gluK5. And finally, just to complete the total number that you could have, you could pick gluK3 and pair that with either gluK4 or gluK5 to create this diheterotetramer. Okay, so those are the diheterotetramers that you can form out of Canate receptor subunits to make functional Canate receptors. Okay, let's now move on to the function of these Canate receptors. The function is pretty much identical to AMPA receptors. They mediate fast neurotransmission. Now, the difference between them and AMPA receptors is that they are much rarer than AMPA receptors. You only find them in decent numbers in certain portions of the brain. So if you're looking at a postsynaptic cell within a glutamatergic synapse, the chances are that most of the receptors on that are going to be AMPA receptors, not Canate receptors. Okay? The other difference is that they don't conduct calcium under any sort of circumstance. Remember, in the AMPA receptors, it was the case that if you didn't have a GLU-A2 subunit within you, you could conduct calcium. In the kainate receptors, it's sodium and potassium which they conduct. But that's not a problem for us, because we know that if you conduct sodium and potassium and you open, what will happen is you will let in a net positive current into the cell, and that will lead to an excitatory postsynaptic potential, which can then lead to an action potential. Okay, right then. So, basically, just to make this utterly blatant, I will draw you a little synapse. So let's say this is the presynaptic neuron here, okay, which will be releasing glutamate, okay, onto the postsynaptic cell here. And in some portions of the brain, instead of having AMPA receptors, you have kainate receptors. Okay, so here is a kainate receptor, let's say. Then what will happen is 4 glutamate molecules will bind to the kainate receptor, 1 to each of the 4 subunits. That will cause the kainate receptor to open. It will then conduct sodium ions into the cell and potassium ions out of the cell. At the normal resting electrical potential difference across the cell membrane, you will get a greater movement of sodium in than you get potassium moving out. That will mean that a net amount of positive charge comes into the cell, which will depolarize the electrical potential difference across the cell membrane, a so-called excitatory postsynaptic potential, which can then trigger an action potential further down the neuron. Generally, the action potential doesn't start within the dendritic spine itself, but the depolarizing current will spread to other portions of the cytoplasm of the postsynaptic cell and then trigger depolarization over there. Okay, right, so that's the kainate receptors then. What I now want to discuss is some agonists for the kainate receptors. So firstly, quisqualic acid, which conveniently we have positioned nicely here, is also an agonist at the kainate receptors. Okay, so it will bind to the ligand binding domains of the kainate receptor subunits and will activate them just as glutamate would, so will cause these receptors to open, causing depolarization of the electrical potential difference across the postsynaptic cell's membrane. Okay, but I want to now discuss the agonist after which the kainate receptors are named, cannic acid. Okay, so let me get a new piece of paper and we'll discuss this. Right, so cannic acid then. Okay, so cannic acid has a pretty simple structure. It's got a 5-membered ring in again, and everything we're doing seems to have 5-membered rings in. Okay, and then you've got a nitrogen at the tip here, so it's 4 carbons and then a nitrogen at the tip. Okay, and all the bonds in this ring are going to be single bonds. Okay, you're then going to have a hydrogen coming off this nitrogen atom here. Then coming out from the front here, and I'm going to stress the optical isomerism here. So it becomes important in this ring because we've got all single bonds. When we had double bonds, it wasn't important because we only had one other group then coming off. But now, because we've got single bonds, which all of these carbons are involved in, it means that each of these carbons is going to have 2 groups coming off. Now, if you imagine that this 5-membered ring is in the plane of the piece of paper, one of the groups that comes off the carbon will be coming out of the page towards us, and the other group will be going into the page away from us. Okay, so this group that I'm now talking about coming off this carbon, it's going to be coming out of the page towards us. Okay, and it's going to be a carboxylic acid group. Right, now there will be another group coming off this carbon, but it will be a hydrogen atom, and it will be going into the page away from us. Now, because it's a hydrogen atom, I don't need to show it because we're drawing a skeletal structure, so that gets me out of that one. Right, now we have another carboxylic acid group coming off, but it's not coming directly off, it's actually a 2-carbon carboxylic acid molecule. Now it's going to come off this carbon here, okay, this one here, but it's going to be going into the page away from us, and the other group that comes off this carbon, which is just going to be a hydrogen, so we're not going to show it, is coming out of the page towards us. Okay, so the way I show bonds going into the page away from us is like so. You gradually draw the lines getting thicker, but you draw them dashed like so. Okay, and then coming off this, you'll then have a carboxylic acid group. So this actually is supposed to be a carbon here, and it will be a methylene group. It will be a carbon with 2 hydrogens coming off it. Okay, and then we've got the carboxylic acid group further back. Now, finally, we're also going to have another group coming off this carbon here, and it too is going to be going into the page, and the unimportant group, the hydrogen, will be coming out of the page towards us. Okay, so here is the bond going away from us, and now we've got a carbon here, which has then got a double bond to another carbon, shown like so. So this is a carbon here, there's then a carbon here which will have 2 hydrogens coming off, so we don't show it. I admit that it looks rather strange in skeletal form, but that's the way it is. Then we have another bond to another carbon which is over here, which is a methyl group. Okay, so can I try and rectify that to make it look slightly less abnormal? Basically, if I draw it out in molecular form, that will probably make it simpler. Okay, this group looks like so. Here's this double bond here between these 2 carbons, and I've got a methyl group coming off here. Okay, and then that's attached like so, and it's going into the page away from us, and the hydrogen is coming out of the page towards us. Okay, so there's that very specific optical isomerism there. Okay, and this molecule then that I've now finished is Cannic acid. Okay, and then the conjugate base of Cannic acid is then Canate. Okay, right. So Cannic acid is also an agonist at Canate receptors, and this is why Canate receptors are called Canate receptors, because they will bind the Canate, and the Canate will activate them to open, just as glutamate would activate them to open. So 4 Cannic acid molecules are going to go in, bind to the ligand binding domains of the 4 Canate receptor subunits and trigger them to open, therefore triggering depolarization of the electrical potential difference across the postsynaptic cell membrane. Okay, right. Next in this little video, what I'd like to turn my attention to is just saying a little line about the delta receptors. Okay, so there are 2 known subunits of delta receptors. Okay, however, although they are grouped with the ionotropic glutamate receptors, and they are actually called the glutamate delta receptors or the delta glutamate receptors, they are not actually glutamate receptors at all. They do not respond to glutamate, and we don't know what their ligand is within the human body at all. Okay, we know very little about them. What we do know is their structure. So let me just discuss their structure. So I'm just doing this for completion, and then we'll move on to the NMDA receptors. Okay, so they are tetramers, okay, and each one of the subunits does have that same basic structure. And I think just for revision, I'll draw that picture again. Okay, so let's draw the phospholipid bilayer here. Okay, then we know the amino terminal domain is extracellularly. So here's the amino terminus. Here then is the amino terminal domain. Then we have the S1 domain, remember here, okay, which will contribute to the clamshell domain. Then the first membrane spanning alpha helix, the reentrant loop M2, the third membrane domain, or the second membrane spanning alpha helix, then S2, which also contributes to that clamshell domain, okay, and then the fourth membrane domain, or the third membrane spanning alpha helix, and then the carboxylic acid terminus. So they all do have this same structure. Okay, so let's just highlight different bits in here to make it look simpler. So here's the amino terminal domain here in turquoise, and I've just smudged that, I think. Oh no, photo felt ink coming off. Never mind. And then we have the S1 domain here in blue, which is in this pre-M1 region here. Okay. We then have our 4 membrane domains. So we have M1, M2, M3, M4. Okay. And then we have S2, which makes the bottom of our clamshell-like domain here. Okay, right, so those are the important domains. And whatever the ligand is for these delta receptors, we would expect it to bind within this clamshell-like domain. Right, okay, so how many different subunits are there for delta receptors? Well, there are only 2 that are known, and these are called GLUD1 and GLUD2. Okay, right, and thankfully, very simply, they only form homotetramers. We have not found heterotetramers which contain both GluD1 and GluD2. Okay, we've only found delta receptors which are homotetramers of GluD1 and homotetramers of GluD2. So both of them form homotetramers where you take 4 identical subunits, stitch them all together, and you've then got a delta glutamate receptor. Okay, and we don't know what they're doing, we don't know what ligands they respond to, we don't know anything more than that about them. Okay, so we'll cast those aside now. They're not glutamate receptors, but they have the same structure as glutamate receptors, so they're called ionotropic glutamate receptors. Okay, right. So in the next video, what we'll do is we'll turn our attention to the NMDA receptors, and this is really interesting stuff now.